Hello everyone, my name is Francesco, and today I will be giving everyone an introduction to biology that is necessary to then completely understand genetics and how genetic diseases affect people. So, okay, so first of all, as a reminder again, sorry for time, but like I would love this to be like very interactive. And the first thing that we need to start to understand is the concept of the central dogma of biology. And all this means is how is information processed and stored in biology? So we all know what DNA is. Essentially, it's the hereditary material structure chemical of life. And so how information flows in biology is that you have DNA, which contains all information about a person, an organism, how they digest a certain food, how tall they are, what body parts they have, and everything. And how the information flows is that you go from DNA, which is a very solid, very stable chemical compound, that is then transcribed, translated into a protein, which will then do the function that the DNA intends it to do. And having a good understanding of DNA is important. And DNA itself, which is the first structure here, is a double helix, which just means it's two strands of DNA that coil with each other. And it's a chemical compound, a molecule that is composed of four molecules. And you might probably have heard these, the four DNA bases. And the four bases, the first one's adenine. Does anyone know what T stands for? I can't see the. Can someone either text in a group, WhatsApp group, or just speak?
I don't I don't know what that means.
Okay, don't worry. Sorry. Okay, so there are four DNA bases, and all they mean of bases is like they are chemical structures with slightly varying structures. They're very similar to one another, but they are different. And the four bases are adenine, thymine, cytosine, and guanine. And they're often in short form just called by their first letter, A, T, C, and G. And what's very important to understand, our DNA, human DNA, animal DNA, whatever, can be essentially summarized with a set of letters. And in fact, when we read the DNA, all we get is like a long stretch of letters, A, C, G, T, whatever the combination is. And the important thing to understand is that these letters together then can compose information that is then used by our bodies to essentially run and make life. And yes. And as I said before, DNA is very stable. DNA, you can see it as a book. It's like a recipe. You can read the recipe, but the recipe itself doesn't do anything. You need to be able to read it, interpret it. And this is where the central dogma of biology comes in. The flow of information from book to action, from book recipe to the product itself, which can be, looking in this analogy, a cake, for example. And we will go more in depth. This entire lecture, this presentation, will go through this process. And the first process is called transcription, which is essentially the conversion of DNA to RNA. So when you have a specific gene, which a gene is just like a stretch of DNA bases, it's read by the transcription machinery to then produce RNA. RNA is another chemical molecule that is quite similar to DNA, but it's much less stable, which is why our hereditary material is not RNA, because RNA is just too unstable, and within I think the half-life is like 20 minutes. Within a very little time, it just degraded and completely like useless essentially. But it's very important in the process of essentially reading DNA and actually interpreting it. And RNA, like DNA, is composed of 5 bases, but to be very precise, one of the bases changes. Thymine, which is part of the DNA, is not present in RNA, and it's replaced by uracil. And just another note, whereas DNA has a double helix, RNA is a single helix. And so then continuing in a process of the central dogma of biology, RNA is then interpreted by by our cellular machinery to then essentially make a protein. And this process is called translation, where we go from a stretch of RNA bases to a protein, which is composed of amino acids. Amino acids are just the chemicals that compose all proteins. And what's very important to understand is that the vast majority of cellular bodily functions are catalyzed, are induced by proteins. Proteins do everything from digesting your food, from composing your muscles, from making you think, and everything. Proteins are very much the functional like component of our bodies. And what's very important to note is that proteins are composed by a set number of amino acids, and these amino acids come together and form structures, proteins that then can do functions. For example, in sickle cell anemia, we're talking about hemoglobin, and in Huntington disease, we're talking about huntingtin. And does anyone know how many amino acids there are in humans? It's not that important of a fact, but humans have 20 amino acids, and the 20 amino acids are used to compose every single protein that we are made of. And it's so here you can really see and appreciate how information flows from DNA to function. So DNA is like the chapter in the book. RNA is kind of like the process of making this, and then the outcome, if you, let's say you have a recipe book, the cake, the pasta, whatever it is the protein itself will then do whatever function it's supposed to do. So now, again, please interrupt me if anything is unclear. I would like to go through this process of DNA to protein and in more in depth. And what I want everyone to think in the back of their mind is that like this is how it's supposed to work in a healthy human being. But obviously, if there are any errors, any mistakes between the DNA to the protein stage, we can start appreciating how this can cause disease. And yes, so a reminder, please, any questions in the WhatsApp group chat because I cannot read the Teams chat. And continuing, now let's talk about DNA. The full name of DNA, it's not important to know what it is, but it gives you context on what it's made of. It's called deoxynucleic acid. So what this means, it sounds like a very complex word. If you break it down, it's not. All this means is that each DNA base is made of an acid that is nucleic, i.e. in the nucleus, and deoxy, it just means it's a form that has one less oxygen. So compared to, so it just means one less oxygen and acid in the nucleus. And as I said before, DNA is an extremely stable molecule that carries all instructions for life. To give you an idea how stable it is, and in the right conditions, we're able to get DNA that is thousands of years old. Like I think in the US, we're trying to like revive the mammoth, and we're doing this by getting DNA from frozen mammoth carcasses found in Siberia. And this comes from 10, 12,000 years ago, just highlighting how stable this molecule can be. And now, I have a question: does anyone know where the DNA is stored in human cells? Any ideas?
The chromosome.
Is it in the
Yes,
nucleus?
exactly, in the nucleus. The chromosome is just the structure that DNA like makes up, but it is in the nucleus. Good job. And so here we see in this diagram on the right, just DNA, and that is coiled up in a chromosome. A chromosome is just a shape in a way, and that is then present in the nucleus of a cell. And so what we can understand is that the DNA is essentially compartmentalized in cells. And okay, And exactly. Okay. And what is very important is that, a very important understanding is that all humans have two copies of every single chromosome. And does anybody know how many chromosomes we have, humans?
23.
Exactly, 23. We have 23 different chromosomes. What this means is that every single one of our cells has 23 copies of every single chromosome. So in total, we actually have 46. And 23 come from our parents, come from our dads, 23 from our mothers. And this is very important because it means that every single gene we have two copies. Let's say we're talking about the gene that digests proteins. We'll have one copy from the parent, from our mothers, one copy from our fathers. So the two copies that are independent of each other. And this is really very important when we talk about genetic diseases. And just restating what a gene is. Here you have a beautiful example, and you have DNA, and then a stretch of DNA, which is just like a certain number of bases that normally can be up from 10,000 to like hundreds of thousands is a gene. In a way, it's just like a functional unit of DNA. And okay, so reiterating, DNA is the one less oxygen version of this acid that is present in our nucleus. Why am I stating important importance of the one less oxygen? Is because, as I said, as I said before, there's another a nuclear acid, which is also very important, RNA, which instead is which is then instead is simply called ribonucleic acid, which is RNA, and that's why it's called RNA. And compared to DNA, it has one more oxygen, which makes it more unstable. And that's we don't have to go in the deep chemistry of it, but it's just an unstable version, which is why it's not used as the hereditary material of life. And talking about the conversion from DNA to RNA, which we talked before, does anybody know what the process of the DNA to RNA is? What's the name for it?
Transcription.
Transcript.
Exactly. Good job. This is the process called transcription, and essentially, here we're going from DNA to RNA. So if we look at the diagram here with where gene is, we're getting a single gene being transcribed, so read from DNA and producing RNA. And if we look at how it works, essentially here we have a nice diagram where we see that the DNA double helix is primed open by the transcription machinery, and then a protein called RNA polymerase. We don't need to remember names, it's just that you understand the concept of it. Is then it then reads the DNA and produces an RNA that is complementary, i.e., complementary to DNA, that is then produced and comes out. Then we can imagine this protein going through the gene and then it comes off. And then the RNA is just a molecule that is produced. And exactly. And RNA is a very versatile molecule. It has multiple functions. But what concerns us the most is a concept of messenger RNA, which is often shortened to mRNA. mRNA, as the word suggests, messenger, is just the form of RNA that is then interpreted by cellular machinery to produce proteins. Okay, and a reminder, if it's anything unclear, please interrupt me and ask. But here we're seeing the conversion of DNA to RNA that is known as transcription. And but then, as before, our final goal is not RNA. Our final goal is proteins. So how do we use the RNA to make proteins? Well, there's the mechanism is called translation. And I know this is a lot of like jargon and scientific words, but once you understand what it is, it becomes very clear, and it's not as complicated as it seems. It's not too scary. But the very important concept is that mRNA is translated into a protein. which, as we said before, is the functional unit in the cellular of life. And so how this process works, which we're going to review later, so don't worry, it's confusing. But mRNA, which is what we just produced from transcription, interacts with with a ribosome. A ribosome is just, you can see it, it's often called like the protein factory. It's like a factory in our cells. And it's what essentially makes proteins from mRNA. And yes, and essentially what how the process is that the ribosome reads the RNA at three bases at once, and slowly it builds a protein that is essentially dictated by a sequence of mRNA. And this is a very key concept, where the sequence of the DNA, which is then transcribed and so makes a corresponding mRNA, is then read, and a corresponding protein is being made. So a set of sequences of DNA bases will correspond to a set of amino acids, i.e. protein. And what's very important to understand is that when we get this conversion from DNA, RNA to protein, it's how the ribosome does it is that it reads three bases of mRNA at once. So let's say we have a stretch of like a very long stretch, the ribosome will interact, for example, here, and as you can see, it will interact with three bases at once. And three bases essentially have a meaning that is one amino acid. And why this is very important is because we said that we have four bases, but we have 20 amino acids. So if we only had one base per amino acid, we can only have four amino acids. But by having a combination, different combinations have different meanings. Like, for example, I don't remember, but let's say AAA is amino acid X. AAT is amino acid B, and so on. And so you can see how essentially we are transcribing and translating information from the DNA to a protein, which is then what will catalyze certain cellular and body functions. And just a very important term is that the three bases of mRNA that correspond to one amino acids are simply called codons. And so what happens at the ribosome, the factory of our cells that makes proteins, moves along the mRNA, adding essentially one amino acid per codon, forming an amino acid chain, which is essentially a protein. So you can imagine the ribosome sliding across the mRNA, ejecting, forming, catalyzing this chain of amino acids, which will fold, forming a functioning protein. And again, just to remember and set the tone, any errors in this process from DNA to protein, if anything goes wrong, we can get diseases. Okay, so this, sorry, now the next, I would just like to re-summarize everything and talk about everything to essentially ensure that we all understand everything. And again, please interrupt questions because now we're just recapping, because I know it's a lot. It's not an easy topic. So as a reminder, oh, sorry, did someone talk? Okay.
Yeah. Hi. I just wanted to ask. Sorry for the interruption.
But don't
Did
worry.
you have any objective for the topics that you presenting, or you just enter into it? Like, is only the central dogma you're talking about? I just want to understand. I came late, so I don't
Yes.
know.
So today, because it's our first session, and we're gonna concentrate on the central dogma of biology. And next session, we'll talk about um, how this impacts diseases. But this is very important because without this understanding, it's hard to understand how genetic disease works.
Yeah. Thank you. Yeah.
And also I know this is like very much from the basics, but like we all have very different backgrounds. Some of us have done biology, some of them haven't, so it's important that we like essentially like set the stage at the same levels, we all understand everything. And continuing, now summarizing, we go from DNA, RNA, protein. This is extremely important to remember. And the next slide is just a video that I found that summarizes everything that we just talked about. Okay, the first step, as we talked about, is transcription, where we get the opening of DNA and the formation of mRNA from DNA. And as you can see, the DNA is being opened, and the cellular machinery is producing RNA, which is this growing purple chain from the DNA. Okay, so and then this part, yeah, it's fine. Yes. And then after transcription, we have translation, where essentially this is more in depth, but we're just looking at the overarching meaning. We're seeing the ribosome slide along the mRNA to make a chain of amino acids, which then makes up a protein. And as you can see, each codon, which is 3 bases, makes up for 1 amino acid. And now the mRNA is gone, and with this beautiful chain of amino acids that will fold, forming a functioning protein. So this is essentially what I would like for everyone to understand. And now following on this, we have a section of questions and Q&A session. So if anyone has any questions, if slides people want me to go through again, please send me texts, interrupt me, and ask me any questions you have.
I have a question. This is just to like kind of understand better. So the DNA itself, without being formed into RNA, is it useless DNA then?
Sorry?
I know this sounds dumb, but like I want to understand. So like when you have the DNA, you said from DNA it moves to RNA, right? And the only
Yes.
way you could do that is to kind of understand the language before you're transcribing. That's how you described it.
Yes, it's
Since like
good.
you're kind of putting it down to understand what it's actually saying before it does what it's saying.
Yes.
So what's reading it is what I wanted to understand.
So what is reading it?
Yeah.
Okay. I mean, that was just an analogy. But what I
Okay.
meant with reading is this exact process of you have
Mhm.
the DNA that is being air quotes read to
Okay.
make mRNA chain out of it. All we're doing is like we are taking the DNA information and transforming into RNA uh, to then process it further. But the key thing is that, as I said before, RNA DNA is stable. RNA isn't, which is why we require this transcription step. Because
Okay.
imagine that like DNA is everything that we are, everything. Height, how we digest, how old we are, everything. And imagine that it's unstable. Then like you wouldn't be able to live. As in like your cells that are dividing would just stop, would like not have some pieces, some genes will be missing, some things will not be there. If that occurs, like you can't function. If you have a cooking book that loses pages, you will stop being able to do some recipes. But if you can't do some recipes, you can't live. So that is why this process is necessary, because DNA is stable and it's essentially it's a record of everything that needs to happen and needs to go through for us to be alive. And RNA is a bit like, you can see it as the cook in a way. It's the thing that takes information from the stable nucleus and then leaves the nucleus to the factories, the ribosomes, to then create the proteins, which would then uh, do the bodily functions that are necessary.
Okay. I actually feel like I understand that better. Okay. So if like the DNA is kind of like the ingredients you don't know what the name of, the RNA would be the specialist that understands how to deal with the DNA so that the DNA can. Okay.
Yeah,
We're getting
exactly.
there.
DNA is just a book. a book with a lot of writing, and it's like, and you don't want the book to change. If the book changes, you may have problems. Like, let's say that you're making pizza and the word for flour changes to uh, I don't know, chili. You can't make you're not making pizza anymore. You make something else. So if the book changes too much, it's just not gonna work.
Understand that better. Actually, thank you for that. Because you might have just done a better job than my science teacher. I understand that.
I'm glad.
Okay.
Any more questions? Please ask whatever, anything. The point of this.
Hello,
Yes.
I have a question. It's kind of similar to what she asked. Normally, if I actually understand, during transcription, you do have mRNA, right, and
Mhm. Yes.
tRNA, and another one which I actually forgot. Is it? I forgot actually. So normally the mRNA is what goes to translation. It's what has been translated to proteins in
Yes.
order to define. So what happens to the other two, like the tRNA and the other one? I actually forgot the name. What happens?
Okay. So there are many types of RNA, but this goes a bit beyond what we need to understand. And okay, but to explain very like shortly, um, RNA is just what is made through transcription, and there are many types of RNA. Different RNAs have different functions. The one that we are concentrating on is called mRNA, because it's a messenger. But then you also have like tens of types. But the most common type, which I'm assuming what you were mentioning, one of them is called tRNA, which I didn't mention because it's not. It's a bit more specific, but it's what you'll see in a second. Its function. And the other type, which I think is what you're trying mentioning, is ribosomal RNA called rRNA. And this goes beyond this lecture, but rRNA is what makes up the ribosome, because the ribosome, it's a structure made of proteins and RNA. It's very complicated, and we don't need to worry about this now. But going back to the tRNA actually is what it's what recognizes, as you can see here, the mRNA, and brings in the amino acids. So the tRNA is the structure here that has three bases, the codon, and then the amino acid attached. But this is not. This is beyond the expectations and objectives of this lecture. But it's a good question. There's a lot to learn.
Yeah, I just want to um add what Mariama said. I think maybe this is just the basic thing, but I think Mariama question also is important because all those three different RNAs, they take part in the translation process. That's
Mhm.
the mRNA and the tRNA and the ribosomal RNA. So
Yes.
that's why he's asking, because you talk about the translational process in it. That's why he was asking what they contribute in it. Yeah, but I understand from the way you discourse it, you just want to, give the background, and then maybe later further discussion you could elaborate
Yeah.
more.
But to quickly like finalize the explanation, so we said that the mRNA is the messenger, which is this. The rRNA is what composes the ribosome. So it's this thing actually, which you can see here. And then the tRNA is the molecule that recognizes the mRNA and brings in the amino acid. So you can imagine the mouse, you can see my mouse, you can imagine this being the tRNA. The tRNA will interact with the RNA and be like, okay, the codon, the three bases, are AAA. AAA is amino acid X. And so the tRNA with the X amino acid and will interact with AAA and essentially add add the amino acid X to the growing amino acid chain. So they're all part of it. And yes, but the most important thing is mRNA. And the reason why is because any mutations in tRNA or ribosomal RNA would just not produce normally a viable organism because we're so fundamental to like anything in what's called in life. That's why like in genetics, genetic diseases, they're less relevant just because they're so fundamental that like no mutations are accepted. And please feel free to ask any questions. Uh, the more questions, the better, because it's best if we, yeah, and yes, that's it. And I'm gonna check quickly the chat here. Sorry for confusing this, but
Another one.
yes.
So the RNA, the messengers, I guess in this case, can you change them?
So
Like you know how do you know how like you can change different DNA structures? Can you change also the structure of the RNA? Because you said it's unstable, whereas DNA is more stable.
mhm.
So RNA, can you change its structure, like genetically, obviously, to like make it more stable? I was wondering if that was possible.
Oh, that's a good question. Yes, there are ways to increase mRNA stability, and it comes down to a set of modifications that can essentially increase its half-life. So obviously, when you're producing mRNA, it will exist in a cell for a set amount of time, and normally that's around half-life is 15 minutes, but different bases, different can increase or decrease how long it's able to survive in a nucleus. So that's true. Yes. But how it's done is quite complicated. We could cover that in an extra lecture, but it's very interesting.
Thank you. Thank you. No, it sounds interesting. Also, it sounds very, I'm not gonna lie, creepy because I think about it. I'm like, so that means you guys can basically find people's DNA and manipulate it in a manner that can create a whole different type of being. Not only just like the way the person is shaped, the way the eyes are shaped, it's kind of scary.
It is scary, but also they're like strong, like especially when you do genetic research, they're very strong, like ethical considerations that you have to like obey by. So you
So you
can't.
guys can't go full Frankenstein. That's what you're saying.
Oh my God, no, 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 no. Uh, Genetic mutations in human cells are you have to be very specific what you do. There's some things you can do, and I would say like literally all research in humans, it's not done on obviously humans. It's done on cells that have been scraped from a person and just and survive on like an agar plate, just like on medium. So so no. But I can see how it's scary, but like ethical considerations are very important in genetics, so don't worry. Any more questions?
Yes, thank you so much. I want to ask about the mRNA. What is the essence of it? Because um,
What is the what? Sorry?
the essence of mRNA. I mean, we have RNA that is um the ribonucleic acid, right? So
Mhm.
we have mRNA that is going to form the protein.
Yes. So do you mind repeating the question? I think I misheard one word. What it stands for? Is that what you're asking?
The mRNA.
Yeah.
What is the essence of it? Because for the transcription, it has to move to mRNA before it could form protein, right?
Yes. As in the essence is just so going back to the central dogma, mRNA is just a subset of RNA. So it's like saying it's a type of RNA. So there are many RNAs, and the ones that are then made to proteins are called mRNA. And the reason why we need mRNA is because we can't directly access DNA to produce proteins, and we need an intermediary molecule that is able to exit the nucleus and interact with the uh, translational machinery, i.e., the ribosome, to then produce protein. So the RNA is essentially is a messenger. That's why it's called messenger RNA, because it transports the information from the DNA to the protein. onto the protein, sorry, to the ribosome to make proteins.
So essentially, the mRNA is like an intermediate, the messenger RNA that is responsible for.
It's responsible essentially for in bringing, transporting the information from the nucleus, where the DNA is, to the ribosome where proteins are made. Because otherwise, what protein will your body make? They will have no way of telling. Like, let's say that you're a child, that you're growing, and you need a growth hormone that tells your body, grow, 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 grow. Then your DNA will say, okay, I need to grow. Let's produce RNA of the growth hormone, which then is transported outside the nucleus and is translated to make the growth hormone. So it's how essentially the nucleus controls the cell and entire body to do anything. In the example that I gave, grow.
Okay, it makes sense now. Man, thank you so much. I get it now.
And please ask any questions, fire away, and at any point. Now, please interrupt me. Now, I would like to start looking at the questions. I've made a couple questions here, which you can see on the left side. I don't want you to read the answers, but essentially, not test, but like stimulate everyone's understanding of the topic, because it's very, I think the best way to learn is to like talk directly about it. So what's very important is the being wrong is good. Being wrong shows that you're learning. So don't feel scared to say something that you think is dumb. There's nothing dumb. Biology is not easy, so we need to, exactly. So don't feel bad. So please, please, please participate and guess. If you get it wrong, it means you're learning, so it's good. Okay, so, and again, any questions, please ask me. But what I wanted to give now is, okay, we learned the concept of the central dogma, but how does it translate to disease, or I want to say disease, but like our daily lives? So does anybody know what lactose is? So
Lactose is the sugar, actually.
yes, yes. Lactose is the sugar present in milk. And so whenever you drink milk, you will be faced with lactose. And so I'm checking the group chat for questions, and yes, perfect. And but the important, and we, okay. So let's say that you are a human drinking milk. And what happens when the milk encounters your stomach and small intestine? How will your body digest the lactose? For lactose, there's a specific enzyme called lactase. Lactase is simply the enzyme, the gene that is responsible for digesting lactose. So I know it's very confusing because the words are very similar, but the O's ending is an ending for sugar, and A's with ASE is the ending for enzymes and genes. So what does it mean that we have a lactose gene? So this is a chromosome, and all humans on chromosome 2, in a section of it, they have a small section that essentially codes for, has information for the lactase enzyme. But this, as I said before, this is a DNA. This cannot do anything to sugar. It's just a DNA. It's a book. It's a recipe. The recipe can't do anything. Now, when we drink milk, we to be able to produce this enzyme, this protein. So to clarify, an enzyme is a protein, is a synonym for protein in a way, and it's simplification. But essentially, what I'm trying to say is, when our bodies are faced with lactose, they will initiate the production of the lactase enzyme gene. Okay, so lactase is an enzyme that digests lactose. And does anybody know what process this is that we're seeing? Where we're having the DNA being opened and RNA being produced from the DNA.
I think it's,
Transcription.
yeah,
Transcription.
exactly. Transcription.
Exactly. That's I was talking about before transcription, and this pink thing that comes out of DNA is the lactase mRNA. So we're seeing when we need lactase, our bodies will express the lactase gene and produce mRNA. Perfect. This is the first step. The second step is now with mRNA, what do we want?
We need
We want a
proteins.
protein.
Perfect. Amazing.
Mm.
Yes. So now we're exiting the nucleus and we're interacting with with this. Okay, I'm gonna ask a question later.
Ribosome.
Right, exactly, ribosome. So again, as we said before, this is translation, and as someone just said, the molecule here is a ribosome, which is just a factory of proteins. And just to summarize, does anybody know what these three bases represent?
They are codons.
Exactly. Three bases represent a single codon, which is equivalent to one single amino acid. And does anybody know what this growing chain from the ribosome is?
So it is actually a polypeptide chain
Yes.
growing from the the transfer RNA.
Yes. So it's a polypeptide chain made of amino acids that makes up the lactase protein in this example. And depending on yes. And then following this, we have the functional lactase enzyme that will will do its function. And in the example, we have lactose, which is a sugar, and the lactase breaks it apart into two smaller sugars. And that is how humans and animals and organisms digest lactose. They produce this enzyme that recognizes lactose and breaks it apart. And so if we didn't have it, this wouldn't work. So can we discussing this, can we all know what lactose intolerance is, but learning about this central dogma of biology, how do people think that lactose intolerance arises?
Oh, is it by the miscommunication from the MRA? Because I was actually wondering about that too.
Yes.
No,
I mean,
yeah,
okay, go.
yeah. I think we may experience lactose intolerance if we actually lack the enzyme, which is the lactase, to break down the lactose to smaller units. I don't know if I'm making sense.
I yeah, I think they are both right actually. It's just that one is specific and the other one is not specific. Like I think the Mariama and was it Sainabou who answered the question. So in other words, the lactase is actually the enzyme that is produced from the template DNA, which will actually help to digest the lactose sugar. So in the case of those who do not have, sorry, those who do have the being lactose intolerant, they definitely will have problem with the enzyme, which will how to call it, disrupt in the digestion of the sugar.
Yes. But one thing to note is that all humans do have lactase gene because I think all children, all babies need lactase to survive because like when we're breastfed, that's what we eat. But most humans lose the ability to produce the enzyme after they're 2, 3, 4 years old. But depending on environment and ethnic makeup, this can be different. And this is why, especially in European populations, lactose intolerance is quite rare because the populations have adapted to continue lactase production throughout adulthood. Normally, in most populations, this is not case, but in some people, this is the case. But for most people, it's more of the lack of the expression, the transcription of the lactase itself. But expanding this, because obviously lactose intolerance is not a disease. It's how do people think that this can be compared to some diseases like sickle cell anemia or other things?
Pardon, can you come again?
No, I'm asking. So now that we've told you this about this example of how lactose intolerance arises, i.e., by the lack of this enzyme, how do we think that the central dogma of biology plays a role in the arisal of genetic diseases? It's more of like a broad question. I'm not asking for like example. It's more like what can go wrong in this process to then cause disease.
So okay.
Oh, go ahead. No, go ahead, Bob.
No, don't.
You get first. Go ahead.
Okay, thank you so much for that privilege. So I think I will just try and make it as short because my other colleagues will want to elaborate. I think if you look at our bodies, for example, a healthy person who's maybe happened to go through mutation, I mean, when his genes go through mutation, it could be maybe some of the how to call it, some of the is it the proteins or the letters are deleted because there is deletion, you know, that could cause a change in the genetic makeup of that person. So if that person happens to maybe by chance, if that person happens to maybe have a child, there's a possibility that that child will actually inherit some part of that genetic disorder. And this is why we can explain heredity of how to call it, genetic disorders. So it was just to explain briefly, but I'm sure my other colleagues can elaborate more. Thank you.
Any more comments on this question?
No, not for me anyway. I think he kind of like touched on what I wanted to say, but he explained it better than I planned to, so I'm good.
It was a very good explanation. It was just like to start to set in the minds of everyone how any errors, any changes in this process will lead to disease. And as it was mentioned, mutations, which are just changes in the DNA, can then affect the mRNA, which in turn will affect the protein that is being made. So if a protein is mutated, so if DNA is mutated, it will change the protein at the end, and that can not always affect its function. And that's how a lot of diseases arise. And this is the point. This is what we're going to talk about in the next lecture. This is just to like kind of like show everybody how this lecture's like objectives link to next lecture's objectives. And so good job, everyone. I think as I was saying things very well. And now going to more multiple choice set of questions. Going back to the very important dogma of biology, how does information flow in biology? So if people want, I think it could be quite nice if every like texted either I guess in the not in the WhatsApp but in the chat here for which do you think is the right order. So you have 4 options: A, B, C, and D. What is the correct order for of the things we talked about today? And also, it's not just order, but it's also jargon. So watch out for transcription, translation, and these words to ensure that you get it right. Some people, yes, have been responding, and indeed, the yes is the right answer. As we talked about, DNA is transcribed to RNA, RNA is translated into proteins. And exactly. So perfect. Good job to everyone. And so let's continue. Now I want to talk more about specifically transcription. So a set of questions, and please respond and yes. Okay, so what does transcription produce? What is the outcome of transcription?
The
RNA.
mRNA, the
Yes,
mRNA.
mRNA and RNA are both right answers. It's just
Okay.
so yes, correct. mRNA is produced from DNA. Perfect. And what is the difference between mRNA and RNA? No, we just mentioned it.
Well, I think RNA is actually how to call it, the template, right, from the DNA after how to call it, transcription. But then RNA is broad. We have a lot of RNAs, right? So the difference here is that there are the RNA, the mRNA is specific. We are when we talk of the mRNA, we are specifically referring to the one that leads to translation, right?
Exactly. Yes.
And when we talk of RNA in a broad sense, we are including all the types of RNA. It could be transfer RNA, messenger RNA, small nuclear RNA, and so on. So yeah.
Exactly. Exactly. So all mRNA is RNA, but not all RNA is mRNA. That's why sometimes they're used interchangeably, but to be completely correct when talking about transcription translation, we should mention mRNA. And that was very well said. Thank you very much. And then last question is all this is more an extension. We didn't talk about this, but is all DNA transcribed? Is our entire genome transcribed?
No,
Yeah,
I don't
exactly.
think so.
No,
You're
exactly.
right. You're right. You're right.
Small sections of our RNA, of our DNA are actually transcribed. A lot of it is very repetitive, and actually, we don't really understand what it does. So, just to give you an idea, wait, sorry, where's the here? To give you an idea, when we look in a chromosome, very little of it will actually lead to proteins. There are around 30,000 genes, and genes that make proteins compose 2% of our DNA. So the vast majority of DNA in humans has nothing to do with this, and we don't really understand what it is. I know it's very annoying, but biology is, we're still learning a lot. Perfect. Okay. The last question is more applied, and it's more just to like give an example that I think people will relate to. So we all obviously know about COVID and the mRNA vaccines. Now that we learned about the central dogma, does anybody want to walk through and describe how we could think that the mRNA vaccine actually works? So what the mRNA vaccine is, you have this, it's called the vaccine itself contains the mRNA, which is why it's called mRNA vaccine, for the spike proteins of the SARS-CoV-2 virus. So, and obviously the when you produce these proteins, they will then be recognized by our body and be like, oh, this is from a virus, let's kill it. So does anybody want to talk about how the central dogma will kind of relates to the key processes that are necessary for the mRNA vaccine?
I want to try. Because in this case, there's already a protein we wanted to create. So would it have to be? It would go into transcribing.
Sorry?
Because I'm thinking like it would have to start with transcribing because you have an RNA already coming in, so you wanted to connect with the DNA. You want to understand the DNA of the individual. So there would be a little bit of transcribing so that the body feels comfortable to take in the proteins that will be created eventually.
I want to interrupt you and
Go ahead.
so it's not exactly like that. It's more simple. Because we're starting with mRNA, we actually don't need to transcribe because transcribe is DNA to RNA. It's one streak. You can't go back. You cannot do RNA DNA. It's only one way. The flow information only goes DNA, RNA, protein. You can't go back. So actually, transcription in this context does not occur because we already have the mRNA. But we're gonna try on what happens next then if we already have the mRNA.
I think the word was translating. I remember the
Exactly.
last. Yeah, because that's the last step. So you're just going straight into the last step.
Exactly. So because yeah, and the reason why you can't transcribe the spike protein is because the protein is part of the virus. We don't have that. We don't have the gene that makes the viral protein. We're not viruses. If you had it, you wouldn't be a human in a way. But so that's why we get the mRNA of a protein we don't have, and it's then enters our cells, and our own bodies then produce by translation the spike proteins. And just to contextualize this, how a normal vaccine works is that they skip this step of mRNA and then just you get injected either the actual virus or the protein itself. So that's where the difference lies. But I thought it'd be interesting just to like contextualize everything, be like, okay, we have mRNA pre-made in a lab, but it's then injected and will yield this protein that we then not evolve, sorry, adapt a response to. And this is the description. Good job. And it's not an easy topic, but I think it's very fun. And so now these were all the questions, and I would like to take any more questions, any more comments, any more, and yeah, anything, because I think it's a good amount of information. It's a lot going on, so I didn't want to go too in depth.
Hello, can you please repeat the procedures that you just the slide that you just passed? I didn't get it pretty much well. Please.
Sorry, do you mind repeating? I couldn't really hear what you said. But what's the question?
Can you please repeat
Yes.
the
So
process?
how the mRNA vaccines work? The mRNA vaccines are this, in a way, orange thing that you see here. You have these containers of mRNA, and the mRNA of the vaccines codes for the proteins, for the viral proteins that we want to respond to. So in a normal vaccine, you just provide the proteins and inject the proteins, and then your body learns this protein is bad. I want to kill this protein, and the virus and the organism that is surrounding it. So what mRNA vaccines do is that they don't give you the protein, they give you the mRNA, which then, as you can see in this diagram, will fuse with our human cells, this big thing, and essentially like pull in all the mRNA of the viral mRNA that is then read and understood by our cell to then produce through translation the protein against which we want the immune response. So now when we see a virus that is similar to this and has this spike protein, our body will know, okay, let's kill it. And it's just a way to, I thought it was a good example to show how the central dogma of biology affects affects our lives in the context of both genetic diseases, but also more, I guess, day-to-day stuff like immune response and COVID. And
Thank you.
yes. And please feel free to ask questions about anything.
Sorry, I just have a contribution
Mhm.
regarding that question about how when we introduce the mRNA in the body. So most of the time, vaccine they are made from like dead bacteria or viruses, but scientists decided to develop this mRNA because of like when you produce the mRNA, it will help to produce the viral protein that's the spike. So
Mhm.
the mRNA vaccine work by like when you introduce pieces of mRNA that correspond to a viral protein, usually the small pieces of the protein found in the virus outer membrane. So they are not exposed to the virus and they cannot be they will not infect with the virus by the vaccine. But by using this mRNA, cell can produce viral proteins
Exactly.
as part of immune response. So the immune system recognizes that the protein is a foreign and produce specialized protein. I think you call them antibodies. Yeah.
Yes.
So these antibodies will help protect the body against infection by
Exactly.
recognizing individual viruses and other pathogens. Thank you. I just wanted to contribute that. Yeah.
Yes. Okay. And so this reminder, I will provide the slides as a PDF form as well with all the answers, because I think it'd be nice if if people had time to go through them again to make sure I understood everything. And please ask me any questions at any point now, later. You can also text me. You have my phone number in the group chat. I can also provide my email, which I probably should have put on the slides. I can do this now. Please contact me for any questions. And sorry, I will add this now for everyone to see. And are there any more questions?